All right, so
Okay.
this is gene therapy and nanoparticles, and we're going to be focusing on hydroxyapatite, and we're going to be referring to it as NHAP. And I am Brooks Maldonado.
I'm Jaden Palacios.
I'm Diego Morales.
I'm Parker Wolf.
All right, let's get into it.
So what exactly are these nanoparticles? Hydroxyapatite nanoparticles, or for short NHAP, is known to be one of the best and ideal gene delivery systems because of its efficient biocompatibility and biodegradability. It is basically the microscopic form of hydroxyapatite, which is a calcium phosphate compound, and is also a mineral that is the main component of our bones and our teeth. Is in terms of the size of the nanoparticles, it varies from around 20 to 350 nanometers. Although there are many different sizes, the most ideal size would have to be around 20 to 100 nanometers. As typically anything less than that has kind of weak nucleic binding, isn't really the best for triggering cell uptake and stuff like that. Anything above 100 nanometers, it makes it too hard to enter the cells. It have the potential to be like trapped by the liver or the spleen, or it can even cause it to kind of accumulate in the tissue a lot more easily. Um, Now, in terms of shapes, it also varies, in which each could also affect the performance of how they interact with the cell membranes. For example, of how much DNA or RNA it can load and how kind of they move and distribute these tissues. As seen in the picture above, the four common shapes would be like spherical, rod-like, needle-like, and plate-like, which is labeled A, B, C, and D. For spherical, it has around 100 nanometer diameter. It is the easiest and it has the most uniform uptake, as kind of the membrane wraps around evenly. For the rod and needle-like nanoparticles, they're very similar as they both penetrate the membranes a lot better than the others, in which their kind of uptake efficiency kind of just depends on their length. So for needle-like, which would be a lot more cell uptake, is it's 200 nanometers long, which is a lot more than rod-like, which is 60 to 100 nanometers long. And lastly, for the plate-like nanoparticles, they have a more complex uptake, but also the highest as they kind of align parallel to the membrane. So they're typically the best for high loading and have a good DNA protection due to their thickness, which is around 50 nanometers. Next slide.
All right, so what is the application of these of NHAP? So the article we chose talked about many different applications. So these are just two of them. And so the first one I'm going to talk about is tumor gene therapy. So one application is tumor gene therapy, and typically the therapy today they use for cancers and stuff is chemotherapy and radiation therapy. But these can damage surrounding healthy cells, which we don't want because it can make the patient sick, and they're also very expensive. Uh, so the high risk and high cost is just not a good option, and NHAP is has enormous potential for a lot of different cancers. And so on the right, I put an image of uh, cancer being treated in a animal test subject. So at the first, we see the no treatment, and the arrow is pointing to the tumor. And by the end, we see a use of NHAP and P53, which is something to fight cancer. And as you can see, it does a very good job at fighting this cancer. And so here, and the second application of NHAP is bone regeneration. So when a patient has damage or fractured or lost bone, the existing therapies like autografts and donors. So like autografts, you'd have to take, it's like taking bone from your person and using it to fix the damaged bone, which you need a secondary surgical site. So it can cause pain, and it leads to a uh, a longer like healing process. And the donor, you need biocompatibility with the donor, and so it can just take longer, and it's just inefficient. But with NHAP cells, as you can see, so this picture is again a picture of an animal, and so the empty cell, you can see the damaged bone, and as we go all the way to the end, we see that the NHAP is does a very good job at fixing the bone, and it just has very good biocompatibility, and yeah, that's why it's good That's why these are the applications for NHAP.
Now, we discussed some of the applications of NHAP, but the question is why is it so effective? First, we need to discuss a few of the important properties. First of all, NHAP is extremely absorptive, allowing the nanoparticles to be easily loaded with nucleic acid. Secondly, the NHAP nanoparticles are very good at protecting the integrity of nucleic acid payloads, meaning that you're not going to have as much possible errors or what have you. Now, NHAP particles effectively release payloads in the cells through the process of osmotic rupture. Within endocytosis, the process in which the cell brings in outside particles, in this case, are NHAP particles containing nucleic acid, the pH in these cells drops due to proton pumps, a natural part of endocytosis. And the NHAP particles respond to this by releasing calcium phosphate ions, which causes the endosomes containing them to burst as water floods in from the cell's cytoplasm, leading to the nucleic acid payload being gently released into the nucleus of the cell. And that is how, essentially, the NHAP particles function. Next slide.
So compared to other nanoparticles, hydroxyapatite is inorganic. So when you have viral vectors, which are like therapy drugs, or even some other non-viral vectors like gold and uh, iron oxide, they can produce cytotoxicity, which damages or can kill cells. And some examples of cytotoxicity is chemotherapy or venom, and you don't want that. So with hydroxyapatite being inorganic, it eliminates the negative factors of organic elements. And with gold and iron oxide, they can get pretty expensive and hard to package. So hydroxyapatite is very easy. It's very cost effective, higher packaging loads. So as a investor standpoint, that can be better to use. And it with what Brooks said, it's very compatible with our bone tissues because of the process of co-precipitation. And that process is an easy way to control the size of the particles that they're injecting and the purity, which can ensure a successful procedure. Next slide. So the source that we used, it was found on Web of Science from the TAMU website. The title is called "Harnessing the Power of Hydroxyapatite Nanoparticles for a Gene Therapy," published on July 6, 2024, written by Dr. Jing. Next one.
All right. Thank you for listening. We hope you learned something. Don't forget,
Nanotechnology
proves
proves
the size
size
doesn't
doesn't
matter.
matter.
All right.